All right, it's my pleasure to introduce Ali. He's a PhD student in the biology department at the University of New Mexico with a bachelor's and master's degrees in veterinary medical science from Alexandria University in Egypt. They dedicated their career to fish disease diagnosis and control. The research journey began in 2016, working with diverse fresh and marine fish farms in Egypt. Currently, as part of Dr. Irene Salinas' lab, the project evaluates different prime and boost mucosal vaccine strategies in farm trout, as well as testing different interval times between prime and boost vaccination. Looking forward to your talk.
Thanks, Nick. Thanks for having me today. I'm going to start with, I would like to start with an old sentence called the protection is much better than cure. That's our main goal of studying and understanding and developing vaccines. As you all know, that there are so many studies on vaccination on humans and animals, but when it comes to fish, we're still in need of more investigation and understanding. And here we can start with some of the importance of vaccination. And sorry again, it's not urgent. Okay, some of the importance of the
Más
vaccination.
tarde, a ver si de pronto le han contestado algo del...
Sorry.
Sorry about that. Somebody was unmuted. I got him.
Okay. So some of the importance of vaccination aquaculture prevents economic losses and fights antibiotic cost and resistance and also secure global food supply. And it enhances the fish welfare and it protects human health against zoonosis disease. Um, and according to the immune response target, we can classify the vaccination routes to mucosal and non-mucosal or systemic. We can start with the most one of the most famous methods is the injectable one, divided into the intraperitoneal or the intramuscular. And when it comes to the mucosal one, we have the emergent route. That is one of the most applicable routes, especially for young fish. Then the oral route, and lastly, the one we are developing and studying, the intranasal routes. So the question is, what we know about the intranasal or the nasal vaccination. Our lab in 2014 showed that there was a high protection level up to 100% against the IHNV if we challenged 7 days post-vaccination, as you see here, and it dropped to 95% if we challenged at 28 days post-vaccination. And this was quite interesting. And it brings us to another question. What if we are waiting longer and using a different kind of pathogens? And are we having the same protection level? So in 2022, we repeated the same experiment with our collaborators, but this time against two different pathogens, the HNV and the Yersinia ricri, the causative agent of the enteric mouth. And the challenge was 6 months post-vaccination. And the result was quite interesting that the protection level dropped after 6 months. So it's recommended to have a boost before 6 months. And actually, that motivated us to go further and try to understand if we boost, when and how it's best for that kind of boost. So this brings us to our goal of study is to evaluate different prime and boost vaccination strategies and to test different interval times between prime and post vaccination in farm trout. And to achieve this goal, we designed our experiment. At the beginning, we chose two different ages before and after 1400 days. Why we choose this one? Because as our previous published paper showed that the lymphoid structure in the nose and nasal cavity, which is the O-node, it's a potential site where germinal center-like structures are formed, first appears with all cell types present at 1400 days. So we designed our experiment to cover the most possibilities for the prime and boost, the post vaccination, and also taking in consideration the availability of a number of tanks in Idaho University where we did the challenge. So we started with 7 groups, priming and priming either one and priming with either priming with intranasal or immersion, and boosting with different kind of intranasal, immersion, and injection. Then we fixed the interval time between prime and boost for both different ages. Then we challenged after 5 months. And as you know, in science, nothing goes the way you want. So, so we did something wrong with the vaccine, with the dose for the challenge virus, and we got LD50 for the 1,000 degree days, the small one, and LD90 for the bigger one, which is 1,500 degree days. That's why we can't compare the survival rate between both groups, but we still have the chance to do more analysis and do this comparison. For now, we are showing the results of the survival probability. The green line in the left shows the survival rate for the control group that hits 50%. And the interesting thing about our results is highlighted in red and blue, and light blue, using the intranasal as a prime or as a boost give us the higher survival rates, more than 95% compared to other methods, and we got the highest protection from the intranasal. For the 1,500 degree days, for the 1,500 degree days, again, at the left side, we can see the green line showing the survival rate was less than 10%, or we can consider the mortality was more than 90% for the control group. But if we focus again on the highlighted colors, the red one for the prime comparison and the blue one for the boost, the comparison here was also can note that the using intranasal as a prime or as a boost is still showing the higher protection, which is more than 80%. And again, the highest protection level was obtained from the IN group. Then we collected serum samples 4 months post-vaccination, just one month just before the challenge. Then we performed the ELISA technique using the specific IgM at deletion of 1 to 160. And here is our results. Shown here, the higher titration in both 1,000 and 1,500 was from the intranasal group, as highlighted with the red color. And also we got significant differences when we are using the intranasal as a post, while priming with immersion, as highlighted by the light blue color. And this is what that was for our first goal. For our second goal, we performed another experiment testing 3 interval times, 4 and 8 and 12 weeks. And again, remember what happens at the beginning. We can't compare the survivability between both ages. So that for the 1,000 degree days, as shown here, for the 1,000 degree days, 95% protection level obtained from 8 weeks group interval, and compared with 90% for the 4 weeks and 75% for the 12 weeks, as highlighted with the red color. And for on the other side, for the 1,500 degree days, the results also was quite interesting, as we got more than 65% protection from the 8-week groups compared with 55% now for the 12-week groups and only 50% for the 4-week groups. Then we had the chance to compare between the antibody titration, and again we got like the 8-week groups interval, the 8-week interval group has the higher titration in both ages, and that was amazing. It aligns with our survival results. And as a conclusion, we can say that for the intranasal route can considered as one of the effective method of the IHNV vaccination. And for the prime and boost combination, of course, the vaccination route is like is important, and the intranasal obtain the highest protection. And for the immersion, if we are going to use it, we need to prime or boost using the intranasal method. And for the optimal interval time, we prefer to use the 8-week interval between the prime and boost to maximize efficacy, and we get like and has like a higher protection, even if the viral load is so high. And for the age and immunity, of course, if we want to make a vaccination, it's better to vaccinate younger because it's going to improve the response and overall protection. At the end, I wanted to thank this project is funded by the USDA, and special thank to our collaborators at Department of Fish and Welfare and Wildlife Science in Ida University, JC, Ryan, Megan, and Veronica, and Luke, and our lab members, Daphne, Ben, Norman, Timmy, Jimena, and Nandini, and Rena is my PI. I want to thank you also. I'm just finished, and I'm happy to receive any kind of questions.
Great job. Thank you very much.
Thank you.
Folks, we have a couple minutes for questions. Feel free to unmute yourself or drop them into the chat. And we have one here, Ali, from Myron Kebis. Can you explain how intranasal vaccination is performed?
Actually, we use micropipette for dropping the solution inside both nares, like in each nare. For our experiment, we use only 25 microliters for each nare and using the IHN cassette. Yeah.
Got it. There's a question here. What kind of vaccine was used?
This is a live attenuated vaccine for the IHNV. Yeah, it was a live attenuated. And we only use the virus one. We never use the bacteria one for now. And we have like some interesting results, and we are working on it. But right now, we just need to show this kind of results. Yeah.
Great. And we have a couple questions here asking about the sort of the commercial potential of this at scale. Is this something that could be implemented?
Yeah, actually, Rena has kind of communication with some companies, and they would just want the results. And once we give them the results, they have like kind of machines and engineers that they can make this possible in the field. Yeah.
Cool.
Yeah.
All right, folks. Thank you very much, Ali.
Thanks for having me, and it was my first presentation here, and thanks for having me. Thank you very much. Thank you.
Well done.